Welcome everyone to the second annual Matter to Life Christmas lecture. Today we have our special guest, Professor Dr. Michael Levin from Tufts University in Massachusetts in the United States. He is head of the Allen Discovery Center at Tufts, and his research focuses on developmental and synthetic biology. I'm sure we will hear more about that in his talk, but as our tradition, we will ask some less scientific questions as part of the introduction. Then we will have around a 30-minute talk from Professor Levin, and then we will take questions. So without further ado, maybe we start with the questions. Um, so, not related to science, but Professor Levin, if you had not pursued a career in the sciences, what do you think you would be doing now?
Well, my original background was in software, so computer science and computer programming. I think I would be a coder. And frankly, up until relatively recently, or maybe it's still true, I always thought that eventually I get kicked out of biology and then have to go back to being a coder. I had this feeling every day of my career trying to get into biology. So yeah, I'd probably just be a coder at this point. But
Mm,
still no doubt doing computational experiments. I don't think I could do without it.
mm, yeah. I think a lot of us would be on a similar boat. Um, Maybe a question now a bit more related to science. So, aside from scientific publications and communicating with scientists, how important is it for you to communicate your work to the general public?
It's very important for me for a number of reasons, and I do quite a bit of open science. I think first of all, it's important for society in general to understand what it is that science does and what the healthy relationship is between science and the rest of society. I think that's very important. Number two, I think it's essential for young people coming up into the field to have access to these things. As you can see here, when I was young, I read many books by people who took their time to explain things and describe the world from their perspective. It meant a lot to me. I think it probably shaped much of who I am. I would like to do that for others. I think everyone should in this field. So I think it's very important. And also, you never know where insights come from. So I get a lot of emails. So I put my stuff out there. I get a lot of emails from people working in psychiatry and trauma and philosophy of language and just all sorts of different fields that normally never touch the stuff that we're going to talk about today. And many of them have really interesting insights, and I think that's really important not to be as siloed as I think things are now. And science communication helps that.
Mm. Thank you. I just want to mention to the audience that I actually found Professor Levin's work through YouTube, so not from a paper he wrote. And I think it also connects to the fact that even as scientists, maybe we're in a bubble, and sometimes you need to escape it. Yeah.
Well,
And so the final question we have, I made this one up, but it's fitting to matter to life. So in this program, we don't really give a definition of life, but everyone, depending on if you're a physicist or a biologist, has to basically have a working definition. So do you have such a working definition for life or living systems? Mm.
So a few things. First, a couple of colleagues and I just wrote a paper, and it's currently in review. But there's a preprint on definitions of life, specifically using machine learning to analyze something like 60 different current scientists' definition of life, and it's quite interesting. Bottom line is, of course, there is no consensus. People focus on very different aspects. I have a very strange view of this, especially for biologists. I don't think defining life is particularly interesting. I think that actually cognition is a broader and much more interesting category than life, and life is a subset of that, and we can talk about that today. But people do ask it a lot, and so I think if I had to say what I think we mean when we say life, what I think we mean are systems that are very good at expanding the cognitive light cone of their parts. In other words, if you have a rock and it's made of the rock constituents, the kinds of behaviors of which they're capable is about the same. Nothing much happened when they were aggregated into a rock. But biology is incredibly good at taking competent subunits, and these are molecular networks, cells, tissues, whatever, and assembling them in a way where the whole is doing things in problem spaces that the parts don't have access to. They have larger goal states. They can solve problems and have memories and goal states and so on that the parts don't have. And I think when we come across systems like that, we tend to say those are living systems.
Thank you for the very insightful answer. I think also if you, like you said, if you asked every single person here, you would get a different answer as well. Um,
Yeah.
okay, so that concludes the introduction. So I will turn off my camera and the stage will be all yours. Thank you.
Great. Thank you so much. Yeah, and thank you for the opportunity to speak to you all. Of course, fascinating title for your session. So, what I'm going to do today is give a talk that I call the unreasonable effectiveness of the behavioral sciences in developmental biology, bioengineering, and biomedicine. That title is patterned on this very famous paper by Eugene Wigner on mathematics and the natural sciences, and I think it's interesting to ask why such a claim, and I'm going to make the claim today that that actually tools from behavioral science are of prime importance to developmental biology, to bioengineering, to cell biology, and that's a very odd claim. People generally don't think that's true. And the reason it seems unreasonable is because we tend to be educated with this sort of ladder of sciences that everybody knows, right? So you've got probably physics on the bottom, and then chemistry, and then biology, and then behavioral science and psychology, and then whatever comes above that. That's the standard view that we're given. I think that's basically exactly backwards. I think that behavioral science goes all the way down, and we'll talk about it. And what I'm going to do is show you some, to basically take you on a tour of data and approaches that others and we have taken over the last 30 years or so to try to explain why this is the case. So the first thing is that in a recent paper, a reviewer wrote. In a review on one of our papers, a reviewer said developmental biologists don't concern themselves with a mind-body problem, and I think that's deeply wrong. Even if they don't know that that's what they're doing, I think that is exactly what they're doing, and I think that if it's not developmental biologists that are going to crack the mind-body problem, I have no idea who it's going to be, because in biology, especially in developmental biology, you have this incredible journey that we all take from a single unfertilized oocyte. Looks to be well described by the laws of physics and chemistry, and then eventually there's this slow gradual process, and eventually something like this shows up, or even something like this. Here's Rene Descartes, and he's going to talk about not being a machine and being something else. And then even after that, some interesting things happen, which I won't get too much into today, although I will show you some anthrobots. And so this is the process of matter, life, and mind. This is exactly where these things come from. I think absolutely developmental biologists have to concern themselves with these issues. And in fact, not only do we take the journey, each of us, from this kind of object to these sorts of things, we also transition across disciplines. So first you're the province of chemistry, and then some physiology, and then eventually, so the developmental biology, and eventually behavior science, and maybe psychology and psychoanalysis, and maybe we take a detour through oncology, and parts of us take a detour through bioengineering. So the important thing about this process is that it's smooth and continuous. There is no magic lightning flash at which point you go from being just matter to being a mind. And so the disciplines are distinct, and that's just for convenience, so that the funders and university departments and journals and things like that can have some sort of boundaries that they try to enforce. But the substrate is continuous. I don't believe it obeys any of the kinds of distinctions that we try to make among these things. And so it's the transformation across this journey that I think we need to understand. My framework is attempting to create a structure that is tools and also conceptual kinds of things, where we can learn to recognize, create, and ethically relate to truly diverse intelligences. And that means familiar creatures like primates and birds and things, or maybe a whale or an octopus, but also some very weird creatures. So colonial organisms, swarms, engineered synthetic new life forms, AIs, whether embodied or purely software. Maybe even someday some exobiological agents, and then also some very exotic things that we're not going to talk about today. I just don't have time. And of course, there are two aspects to trying to do something like this. First of all, people have tried before. So this is Rosenbluth, Wiener, and Bigelow back in 1943, trying to show what the great transitions might be on a scale from passive matter to human level metacognition. And what I always have in mind is this electromagnetic spectrum. this idea that before we had a good theory of electromagnetism, we had magnets and light and lightning and static electricity. And we thought those were all different things. We had different names for them. We thought they were different things. And because of our own evolutionary history, only a tiny bit of that spectrum was accessible to us. But what a theory enabled us to do was, first of all, have a unification, where in fact we now understand that all of these things are part of the same phenomenon. And it allowed us to make technologies to let us operate in regions to which we are normally not able to reach. And so I think something very similar is going on here in science, where we have a tremendous amount of mind blindness. That is, because of our own evolutionary history, we're only good at recognizing intelligence in very narrow types of embodiments. And so what I'd like to do is to create tools to ameliorate that. And in particular, in ways that are not just philosophy or linguistics in terms of redefining intelligence or anything like that. I want to move experimental work forward. I want this framework to drive new biomedical discoveries, new capabilities in synthetic morphology, and of course, better ethics. And if you want to read a very lengthy exposition of that, this is a kind of a 1.0 version of that. Okay, so one of the things that I always think about in trying to put different types of agents on the same scale is what do they all have in common? And I think one way we can start to think about this is through the axis of persuadability. In other words, as engineers, you look at a system and you ask yourself, what tools can I use to optimally interact with that system? Maybe hardware rewiring is the only thing that I can do, but maybe the tools of cybernetics and control theory will do, because we're talking about some kind of homeostatic system, or maybe training with rewards and punishments, so behavioral science, maybe psychoanalysis and friendship and love and whatever else goes on here. So what I think that any kind of a claim about a system is really a protocol claim. What you're really saying as an engineer, you're really saying, here's this bag of tools that I plan to bring to that system. Let's all see how that works out for me, and then we find out. So that means it's an empirical question as to how much or what kind of intelligence any system has. You can't decide it from a philosophical armchair. You can't try to prop up ancient philosophical categories about what intelligence or cognition means. You have to do experiments, and you have to see how they work out, and then we can all see how that went. Now, this is the sort of thing we're made of. We are made of an agential material, meaning that unlike wood or metal or these kinds of things that basically just hold their shape, we are composed of, for example, cells. So this is a single cell called the lacrimaria. There's no brain. There's no nervous system. You can see it has this like incredible ability to get its goals met in the local microenvironment. If you're into soft robotics, you might be drooling right now because we don't have anything remotely this capable that we can build. We talk about, Jamie Davies and I talk about how you engineer with these kinds of materials, which is very different from engineering with normal matter. And so in biology, we actually have a multi-scale competency architecture, because all the way from the molecular networks up through cells and tissues and organs and whole animals and swarms, all of these systems are not just scales of size, but they're actually different. They have different competencies projected into different spaces. And what I mean by that is simply that people are very used to thinking about intelligence as movement through three-dimensional space. So when you see crows picking up cigarettes to receive a reward or something like that, it's a medium-sized object moving through medium speeds and 3D space. We're primed by our evolutionary origins. We're primed to recognize that. But biology has been doing that kind of sensing, decision-making, actuation loop in many spaces long before we had muscle, long before we had nerves. So lots of biological materials navigate transcriptional spaces, high-dimensional, difficult landscapes like that, physiological state spaces, and anatomical morphospace. This is the one we'll spend the most time talking about today, which is the space of anatomical possibilities. So what we're interested in is understanding this navigation. What kind of information processing do these systems do? What do they know about their world? How much memory do they have? All those kinds of things. So I'm going to use, for the purposes of today, I'm going to use one definition of intelligence. There are many. I'm not claiming this is the only definition, and I'm also not claiming that it captures everything. It doesn't, for example, capture play and exploration and things like that. I'm going to focus on one particular aspect of it, which was emphasized by William James, which is some degree of the ability to reach the same goal by different means. This is nice because it focuses on a measurable thing, that is the ability to reach certain goal states under different circumstances. It doesn't talk about brains. It doesn't talk about a specific problem space. It's a nice general cybernetic definition, very helpful. And so the middle part of this talk is going to be basically just exploring the data that we've generated with this hypothesis, that morphogenesis, so pattern formation out of cells in the vivo or in vitro, is a collective intelligence. That collective intelligence, it's like all of us, it's made of parts. All intelligence is collective intelligence. It exerts its behavioral competencies in an unusual space, an anatomical morphospace. And it's a nice stepping stone towards understanding really diverse or unconventional intelligences, because it's related to us. It's made of the same stuff, so it's not completely alien. And yet it operates in a different scale with different kinds of goal states that it can undertake in anatomical morphospace. And it really significantly stretches our imagination and our ability to generalize from our kind of ancient pre-scientific experience. So I think this is a very valuable exercise. So what we're going to look at next is some examples of what kind of collective intelligence do cellular swarms deploy. And what we're going to see is that it works on a different time scale. So instead of minutes, we have to, instead of milliseconds in brains, we have to think about minutes and hours. It's a different different problem space. So not three-dimensional motion, but changes in morphospace. Interestingly, same mechanisms, same exact mechanisms as your brain uses to do this, ion channels, gap junctions, neurotransmitters. We'll talk about that momentarily. And many of the same algorithms, the tools of cognitive science work great in this field. And actually, Thomas O'Brien developed this amazing tool that you can go to this website called FieldShift, which automates something that I used to have my students do manually, which is to take a neuroscience paper and just replace words. Every time it says neuron, I say make it say cell. Every time it says milliseconds, say minutes. And if you do that replacement, generally you have yourself a pretty reasonable developmental biology paper, because there is incredibly deep symmetry between the question of where bodies come from and the question of where minds come from. So let's just look. So I'm going to take you very briefly through these examples. And if you want to see the papers and the underlying data, everything's on our website. So we're just going to run one by one through a bunch of established components of intelligence. First, goal-directed activity. Okay, so here's a simple example. Here's an axolotl. We know that it is not simply a matter of feed forward open loop emergence, how you get from an egg to an axolotl, because if you amputate the limb, then the cells will very rapidly grow, they will pattern, and then they will stop. That's the most amazing part, is that they know when to stop. When do they stop? They stop when a correct axolotl limb has appeared. And so this is a context-sensitive homeostatic system. You can think about it as trying to keep to the same region of anatomical space, this particular region. If you're deviated from it, you'll do your best to try to get back there. So this is not simply a progressive unrolling of some kind of cellular automaton or something. This is very much context sensitive, and it has a set point. We know it has a set point because we can see it. And if we deviate from that set point, that is the state towards which it will come back. Now, another interesting thing about intelligence is not simply that it can reach the same goal every time, but it can do so by different means. And so one simple example that I can show you is the transition from tadpoles to frogs. So tadpoles have to rearrange their face. So the eyes, the nostrils, the mouth, all of these structures have to rearrange. So you get from a tadpole to a frog. And that was thought to be a hardwired process. I mean, all tadpoles look the same. All frogs look the same. If you do the same motions in the same direction, you should get from here to there. So what we created were so-called Picasso tadpoles. We scrambled the face. So the eyes on top of the head, the mouth is off to the side. Everything is completely scrambled. And what we found is that basically these creatures make pretty normal frogs, because all of these structures don't just go in the same pattern they always go. They move in novel ways. In fact, sometimes going too far, having to come back. They have all kinds of rearrangements to get to the correct final state. And we're going to talk in a few minutes about how do they know what the correct state is. We'll talk about that. But what the genetics gives you is not a hardwired set of movements. It actually specifies a program that executes a flexible ability to recognize unexpected states and take corrective action toward a specific goal. So this idea that we're all told in chemistry and cell biology classes that none of these systems know anything. Well, we somehow know things, but cells and tissues definitely don't know anything. They just sort of follow hardwired rules, and eventually interesting things happen. So that's a claim. That shouldn't be an axiom. That should be an empirically testable claim. The claim is that open loop emergent, so like cellular automata, things like that, that those kind of models are going to be sufficient to explain and harness morphogenesis. And it turns out, as a matter of empirical testing, that claim is incorrect. Those are useful models in some cases, but they definitely don't give you everything you need. There are many other kinds of tools in the study of goal-seeking systems, so cybernetics, psychology, and so on, goal-seeking systems that are very useful in our field. You should not think that there's some sort of rule that you have to stick to open loop kinds of models. The next thing that's really interesting and relevant to what I was just saying has to do with hierarchical non-local control. So I want to point out this experiment done first in the 1950s, when you take a tail and you graft it to the side of an axolotl or a newt or some other kind of amphibian. So what happens then is that over some period of time, this thing metamorphoses into a limb. In other words, the tail turns into a limb structure. It remodels. Now, pay attention to the cells at the tip of the tail. They eventually turn into fingers. Why? There's nothing wrong at the tail. There is no damage. There's nothing wrong here. There are tail tip cells sitting at the end of the tail. Why would these things suddenly start turning into something else? It's because there is a kind of very important top-down control where the parts are aligned towards a goal that they don't know anything about. Individual cells have no idea what a tail is or what fingers are or anything like that, but the collective absolutely does. And you know it does because if you deviate the large-scale form by putting a tail where a limb should go, over time the system will correct to a large-scale target morphology that is more correct. In other words, having a limb here instead of a tail. And in doing so, it filters down all of the control signals, filter down to change the molecular biology of the cells at the tip of the tail, even though locally there's nothing wrong, to turn them into fingers. So while the individual cells have no idea why they're being reprogrammed and remodeled and killed off and so on to become these kind of fingers, the collective is doing what collectives always do, which is to deform the option space for their parts so that the parts will then do things that are aligned with goals of the large scale in some other space that the parts don't need to know anything about. Notice the interesting similarity between voluntary motion. So every morning when you wake up, you might have very abstract research goals, social goals, financial goals, whatever. These are incredibly abstract kinds of things. But in order for you to act on them, ions have to cross your muscle membranes. Like this is the essence of mind-matter interaction right here. Your goals and your hopes and dreams and your plans have to eventually be able to make the biochemistry do things in your muscles, right? And your whole body, using the electrical system we're about to talk about, your whole body is a set of transducers that allows that to happen. So for the exact same reason that the ions in your muscle membranes dance due to high-level abstract goals in weird spaces that you might have, for that same reason, these kinds of molecular events inside these cells are obeying a large-scale anatomical plan. Okay. One other interesting component of intelligence is hackability. That is, intelligent systems generally have a kind of software, not just hardware that they're made of, but they actually have a kind of software that can be exploited to get them to do things they don't normally do. And in order to look at that, this is now where we're going to talk about bioelectricity and the interface that my group uses to access the reprogrammability of life. So let's first ask, where do anatomies come from in the first place? So here's a cross section through a human torso. Lots of amazing complex structures. Where was this pattern specified? Okay, most people immediately will say, well, the genome, of course, it's in the genes, but we can read genomes now. We know that none of this is directly in the genome. What the genome specifies are proteins, so the tiny molecular hardware that every cell gets to have, and some timing information around where certain things will be expressed. But this is all the function of the software that's encoded by that molecular hardware. The information of this kind of information isn't in there any more than the structure of termite colonies or the specific structure of spider webs is in the genome of those species. So we need to understand how does this work? And what I'm going to tell you is that it works very similarly toward as in another system that we all know, which is the brain. So in the brain, the hardware is made of ion channels. So these are proteins that let charged chemicals in and out. So calcium, potassium, sodium, chloride, that kind of stuff. In and out, and because of that, it acquires a voltage gradient, and it can pass on that voltage information through electrical synapses onto the neighbors. And the software, and here's this group did this amazing video of a zebrafish brain, of a living zebrafish brain doing whatever it is that fish think about doing. That the cognitive content, right, the memories, the preferences, all of that stuff is encoded in the electrophysiology of this network. So that's the commitment of neuroscience, right? Neural decoding is this idea that if we were able to read and decode these electrical signals, we would know what the creature is thinking about, what its memories are, and so on. Well, okay, so this is an amazing system, but it turns out that it's incredibly ancient. This is not really about nerves and muscles. In fact, I don't think neuroscience is about neurons at all. It's about this kind of cognitive glue, the processes that make us all a collective intelligence. It's why you know things your individual neurons don't know, because it makes a computational network that can operate in other problem spaces. And so biology discovered this a very long time ago, so around the time of bacterial biofilms. And so now every cell in your body has ion channels. Most cells have these electrical synapses to their neighbors. And we have been running that same kind of decoding program, just not in neurons, looking at embryos to ask, what did these electrical networks think about before there was motion such that you could think about positioning yourself in three-dimensional space? So to do that, we developed some tools. So back in 2000, we developed the first molecular tools to read and write electrical information into tissues, into non-neural tissues. And so we have voltage-sensitive fluorescent dyes, and we can do this in vitro. So the colors here reveal voltage. These are not models. These are real data. The colors reveal voltage in this early frog embryo. You can see all the conversations that the cells are having with each other, and lots of computational tools to try to understand all the way from the molecular networks that express channels up to tissue properties of what happens to these electrical patterns, and then how they do things like pattern completion, aka regeneration, how they store memories as attractors in these electrical networks, and so on. So pattern memories. I'll show you just really quickly what they actually look like. So this is a video of what we call the electric face, defined by Danny Adams in my group years ago. This is well, there's a lot going on here, but here's one frame from that video, and you can see there's a bioelectric pre-pattern. Here's where the eye is going to go. Here's the mouth is going to be here. The placodes are out there. This is well before the genes come on to pattern the face, and if you change this electrical pattern, the pattern of the face changes. Not only is bioelectricity the memory medium that helps complex structures know what they should look like, it also works not just to glue individual cells into large-scale structures, but actually across embryos, so multiscale. So here's an example. We poke this embryo, and you can see within minutes, all of these other guys find out about it, right? So it's a kind of multi-scale kind of thing. So here, if we poke this one, these two, even though they're not touching, right, that information spreads. And in fact, we now know that groups of embryos form a kind of hyperembryo that can solve problems better than individuals. In other words, large groups resist teratogens better than small groups. They have unique gene expression and so on. So again, you see this idea of bioelectricity being able to align competent parts into larger scale systems. So now we're going to get to this reprogrammability angle. So having seen this, and having seen this idea that bioelectricity is how cells normally communicate with each other to make all kinds of patterning decisions, we wanted to try to exploit that. And I want to show you one simple example. This is a video which I'll play momentarily. Here are the before and after frames. Again, the color represents voltage. And so here it is. You can see this cell, before it touches this cell, it has a different voltage than all of these guys. But as soon as it touches, it has the same. So here's the video. So it's crawling along, minding its own business. This guy is going to reach out, touch it, bang. That's it. Now it's green like the rest of them, right? So the ability to inject an electrical signal through even an extremely tiny contact like this has the ability to change the cell voltage and change the cell behavior. So we wanted to we wanted to be able to do this as well. And so we developed tools. Now, we don't use any electrodes. There are no magnets, no waves, no frequencies. We don't do anything like that. We use all the tools of neuroscience. In other words, we manipulate ion channels and gap junctions. That means neuropharmacology. It means optogenetics. It means being able to mutate these channels, open and close the gap junctions, right? So we can control the electrical state of these cells, and we can control who talks to whom. And so I'll just show you one of many examples, actually two examples, for what this means. So here's the side view of a Xenopus laevis tadpole. Here's the gut. Here's the animal's right eye. Here's the mouth over here. The brain is up here. What if we inject an ion channel encoding RNA encoding a particular ion channel into cells that are going to become these gut cells here? What we can do is set up a little voltage spot that looks just like that eye spot in that electric face that I showed you. And so what was done here by Vaibhav Pai and Sherry Au in my group was to inject the ion channels, cause the voltage spot to be there, and the cells interpret it as a make an eye signal. And sure enough, they make an eye, and here it is. And those eyes have lens, retina, optic nerve, all the same stuff. And notice two interesting things. First of all, it's instructive, but it's very modular. In other words, we didn't have to specify how to build an eye, right? That's the benefit of dealing with an agential material, is that you don't micromanage it. You give it cues or prompts. So we gave a very simple voltage signal that said, make an eye here. All the downstream stuff, all the regulation of stem cells and morphogenesis, everything was taken care of after that. Exactly what happens in cognitive systems. When I'm talking to you now, I don't need to worry about putting your synaptic proteins in particular places. You'll do that yourself. I'm just giving you text prompts, right? So that's the joy of communicating with a multiscale agential material. And I'll also note that the blue cells, so this is a cross section through an ectopic lens. The blue cells are the ones we injected, but there's tons of others that are contributing to this morphogenetic process. We didn't have to tell them to do that. We didn't have to warn them that there's not enough injected cells to actually make an eye. They will do that themselves. They recognize that there's not enough. They recruit their neighbors. There's actually a bunch of back and forth because the neighbors resist, because this is also how tumors can form. So the neighbors don't immediately snap into line, but when things go well and the signal is sufficiently convincing, which is what we want as workers in regenerative medicine, we want to be able to motivate these cells. They will do all kinds of other things like recruit their neighbors and undergo morphogenesis and so on, right? So prompt, not micromanagement. One other example I want to show you is planaria. So this is regeneration. Here's what these worms look like: one head, one tail. And when you cut them into pieces, they reliably, every piece, so let's look at this middle piece, it reliably makes one head, one tail. So you might wonder, how do they know how many heads they're supposed to have? And so we looked, and it turns out that there's this voltage gradient that says one head, one tail. And what we can do is take this animal and reprogram the bioelectrics, and this is again done with drugs, with ionophores and ion channel drugs, and we can establish a different state. So two heads, and you can see it's kind of messy. The technology is quite messy still, but what will happen then is that the anatomy is still quite normal. The molecular biology is still quite normal. In other words, the anterior markers are expressed up here, not in the tail. Okay, so the molecular biology doesn't know anything's happened. The anatomy doesn't know anything that's happened. It's a latent pattern memory. But if you then cut this animal, this is what it does. It in fact will go and make two-headed worms. Then this memory becomes active, and it will in fact do what the memory says. So one normal planarian body is able to store two different representations of what a correct planarian should look like. And that representation is a counterfactual memory. It's not true right now because I only have one head right now, but it is what I'm going to do in the future if I get injured. So you can see what's going on here. This is the pattern memory. If you want to know how all these systems know what they should build, they have a bioelectrical encoding that they maintain of what the correct pattern is, and then they will simply try to reduce error towards that encoding, but it's rewritable. And the reason I keep calling it a memory is because it is stable, but it's rewritable. And so if I take these two-headed worms and I cut off this ectopic secondary head, it doesn't go back to normal, even though the genetics are completely normal. We haven't touched the genome. It will continue to make two-headed worms because that's where the memory is. It is not genetically specified. What the genetics does specify is a hardware that by default acquires a voltage memory that says one head, but it's totally changeable. And we can change it back. We can take two-headed worms and change them back. And here you can see this is a video of what they do when they're just hanging out. So this question of where is the fact that they should have two heads, where that's stored, it's subtle. The genetics matters because you need to have hardware that's capable of acquiring those states. But after that, it is actually stored in the bioelectrical state, and you don't need to change the genome any more than we need to change our genomes when we learn new things in the brain. Now, not only can you reset this animal to have two heads, you can also ask it to have heads of other species. So we can take this triangular form and we can ask it to make flat heads like a P. felina or round heads like an S. mediterranea by perturbing the bioelectrical pattern memory that encodes which of these attractors in anatomical space they normally go. These other species live in these attractors, but the hardware is perfectly capable of visiting them if you push it in that direction. And the distribution of stem cells and also the shape of the brain becomes just like these other species. Something between 100 and 150 million years of evolutionary distance between this guy and these actual things, but no problem traversing the morphous space. In fact, you can ask them to do really much more kind of exotic things that you can go further in that latent space that aren't even flat planary at all. So you can make these spiky things, you can make round cylindrical things, you can make hybrids, and plants do this too. This is pretty much the only thing I'll say about plants in this talk, but they absolutely have this kind of plasticity. And you can see that the reliability of development is really obscuring a lot of important things, because we see these acorns and we know that they very reliably make these oak trees, and we think that that is what the genome knows how to do. The oak genome knows how to make these oak trees, these oak leaves with a very specific shape, right? And the reliability of it hides the fact that actually, if prompted by a really good bioengineer, and these wasps are amazing at putting down prompts that actually cause the plant cells, they don't build these themselves. The wasp isn't building this. These are plant cells. They make these galls with these incredible structures. We would have absolutely no idea that these cells are even capable of doing this. But the plasticity and the other aspects of the morphospace are revealed when you start probing it. Okay, and so bioelectricity is how we do that. And so just the last couple of things I want to show you, of course, learning. And so you might be surprised to know that even molecular networks, never mind cells, but the molecular networks inside of cells are capable of 6 different kinds of learning. So association, associative conditioning, habituation, sensitization, just from the network itself. You don't need special proteins for this. It's just the properties of all kinds of networks can do this, and we're taking advantage of this for drug conditioning and other applications. Large scale memory. This is an amazing phenomenon called trophic memory in deer antlers. So discovered by Bioengineering, where if in 1 year, if you make an injury, the whole you get a little callus, and then this whole thing falls off. Next year, they grow a new rack, but it has an ectopic tine at the location of last year's injury. Okay, so the system has to remember where the damage was, store the information in the stem cells or somewhere, and then when it rebuilds the bony structure, to take a detour in that same location. Think about what molecular pathway model you could possibly draw for something like this, right? These kinds of arrow diagrams that we have in all our cell biology papers. Like, it's incredible. We don't really have good tools yet for dealing with things like this. Not only that, there are aspects of creative problem solving. That is, sometimes you can't get to the goal that to the target state by your normal means, and so this is the kind of example where if a kidney tubule is normally built by a large number of small cells working together, if you make the cells gigantic, no problem, it will reduce the number of cells, but eventually one single cell will bend around itself and still give you that same large scale structure. Different molecular mechanisms. This is cell-to-cell communication. This is cytoskeletal bending. Use the tools you have to solve the problem in a novel circumstance. My cells are too big. I have tools that I can use to get here. That's the component of every IQ test. And so we use these kinds of insights to address birth defects. So here's a really severe brain defect in a tadpole caused by a mutation of the Notch gene. So here they are bearing the mutation of the Notch gene, but we have a computational platform that told us how to give them the appropriate bioelectrical pattern memories to get them to rebuild a correct brain, even though the notch is still mutated. So some hardware defects are fixable in software. If you understand the pattern that is here and what the cells are doing, you can then ask them to build new structures, very complex structures, again, without micromanagement. In the same vein, we have a program on regeneration. So frogs, unlike axolotls, do not regenerate their legs. But what we were able to do is to provide a bioreactor with a bioelectric payload that basically triggers pro-regenerative genes and growth cascades. And here by 45 days, you've got some toes, you've got a toenail here, eventually a pretty respectable leg. These legs grow for a year and a half after the intervention. Our intervention is 1 day, 24 hours. After that, the legs can grow for a year and a half, and we don't touch it during that time. And again, that's because we're not trying to micromanage cell or molecular events. We're trying to signal to the system to commit it to the leg building path instead of the scarring path at the beginning. And then the final thing I'll show you is our work on cancer. And so the idea is that when cells disconnect electrically from these pattern memories, they roll back to their evolutionary precursors, and they basically become like unicellular creatures as far as they're concerned. The rest of the body is just external environment to them. They go where they want. They reproduce as much as they want. And we can detect this non-invasively with the bioelectrical imaging. You can see before that's going to happen. And then if you artificially force them to remain in connection with the other cells, you don't fix the oncogene, you don't kill the cells, no chemotherapy, but you control the bioelectrics, then even though the oncoprotein is blazingly expressed here, in fact, it's all over the place, this is the same animal, there's no tumor. And that's because the outcome is not directly a matter of the genetics, the outcome is a matter of the physiological decision-making that happens. And the final thing I want to show you is this notion of novel beings. So everything that I've shown you so far is creating evolutionarily provided structures. So normal frog eyes, normal frog legs, frog brains, all of that. What happens when we move to novelty? So this little creature, which looks like something I might have found in a pond or a lake somewhere, if you were to sequence the genome, you would see 100% Homo sapiens. So we call these anthrobots. We allow them to self-assemble from human adult, not embryonic, adult human tracheal epithelia. They put themselves together into this little motile creature. It has all kinds of interesting capabilities. For example, if you put them down on a bed of human neurons that you've put a big scratch through with a scalpel, you make a wound, they will assemble into this kind of superbot cluster, and they will start to knit together the gap. This is what it looks like when you lift them up. They start to repair. Who'd have thought that your tracheal cells, which sit there quietly in your airway, have the ability to be a self-motile little creature? This is what they look like with the little cilia on top. This is how they swim around. They have over 9,000 differentially expressed genes. So they have their own transcriptome that's about half the genome is different. We didn't touch the genome. They have exactly the same unedited human genome. There are no synthetic circuits, no transgenes, no weird nanomaterials. But what they do have is a different lifestyle. This is releasing native plasticity, and they have 4, not 12, not 1, but 4 discrete behaviors that they can transition through. And the one thing I didn't show you, which is what all this is, which are xenobots, which is similar idea made from frog epithelia. But the question I want to ask is this: xenobots and anthrobots have no straightforward evolutionary backstory. There have never been any xenobots or anthrobots. No anthrobot has ever been selected for the ability to swim or to repair neural defects or any of the other things it's doing. Where do their properties come from? If it's not from a story about past selection forces, where does it come from? And we know when the computational cost was done to design a human or a frog, it was done in the eons of that genome bashing against the environment and being selected. When did we pay the computational cost to make good xenobots and anthrobots? And I don't have time to really go into it, except to point out that there's some really exciting work from a number of people, and you can see all this at our symposium. This is an asynchronous symposium on the Platonic space, with the idea that the latent space of mathematics provides some very important information that impinges not only on physics, but also on developmental biology and on cognitive science. And so there's some interesting things to be said about the mind-brain relationship, and I encourage all of you to come and check that out. And so the very final thing, I'm running out of time here. The very final thing I'll point out is our efforts to sort of breach the 2 areas that I was talking about today. So I was talking about the plasticity of of life itself and the development of cognition in minimal media, and our tools to study that. So what tools and apparatus do we have to explore that space? And I want to kind of merge those two categories and show you our latest piece of engineering, which is a robot scientist platform that basically what it does is it makes hypotheses about the kinds of signals that have to be given to cells to get them to do whatever. So it makes hypotheses about anatomical morphospace. It then carries out the experiments, observes what happened, learns and changes its hypotheses, and so it does this kind of loop, right? So the idea is for it to learn to make xenobots, anthrobots, and all kinds of synthetic bioengineered constructs to specific goals. Now, we've had this diagram as a kind of sort of hopeful vision of what we want for the longest time. But what I can tell you now is that we've now got the thing running in our lab. What it's doing is it's exploring the space of prompts for cellular collectives because it has components by which it can produce chemical stimuli, electrical stimuli, temperature stimuli, vibration, other kinds of ways to communicate with the material. And then what it has is it has both a brain and a body. So the brain is a set of AI agents that try to compete for better ways to understand that space, and then it carries out experiments. This is what the thing actually looks like standing in our lab. It's only been operational for a few weeks, so these are very early days. This is an extremely early version of this, but this is, to my knowledge, the first robot scientist that operates in morphospace. It's not doing experiments on metabolism or things like that. It's actually building new biological structures by exploring that space. So it is in fact an embodied AI. The AI has a body. It doesn't move through three-dimensional space. This thing sits still. But what it does do is navigate morphospace, and it navigates the space of anatomical possibilities via a living front-end interface, which happen to be, in this case, frog cells. And so the future impacts here, and I'm wrapping up here. The future impacts are first the obvious thing. So useful synthetic living machines. If we can get that cycle of discovery sped up, that means we can do in-the-body repair with anthrobots made of your own cells, environmental cleanup perhaps with xenobots, exploration of maybe space and so on. Sort of going one step deeper, this is a robot scientist that can help us crack the morphogenetic code. If we understand how to communicate new goals to groups of cells, that leads to new regenerative medicine of all kinds of interesting applications, and of course, bioengineering towards the notion of an anatomical compiler. That is arbitrary, being able to specify arbitrary forms, right? And we're hoping that the next generation of this kind of system will help discover that. But even going one step deeper and kind of weirder is this idea that what this really is a tool to communicate with unconventional minds. And you can think about it two ways. You can think about it as a tool where the human scientist, through this MOMBOT interface, MOMBOT is this thing that I just showed you, can actually interface to the collective intelligence of cells, right? We specify our goal states, the MOMBOT figures out what stimuli those have to be and gets them to execute, exactly as your high-level cognitive processes issue commands that eventually through your body have to get molecules to act in your muscle cells so you can carry them out. But another way of thinking about it, actually, and I think these are both useful, is that the MOMBOT is the active agent, the frog tissue is the translator interface, and what it's exploring is the space of possible forms and functions. So there are different perspectives on this. You can take the perspective of different parts of the system, and I think all of these are important because in the future, right, it isn't only language models and standard AIs that you have to think about. This kind of plasticity, right? The fact that we are all going to change both biologically and technologically, slowly and gradually, because life is interoperable. Every layer can be interacted with synthetic engineered components, and eventually this is going to be your neighbor, right? It's not going to be as easy as, well, I'm a real biological and this is just a machine, it's a language model, I think we're different. No, this is what we're going to be talking about is really stretching the notion of what an active agent is, and the fact that you do or do not have components that were evolved by trial and error over millions of years, or components that were designed by engineers. Those things are going to mix in a way that make ancient categories completely irrelevant. And so this is sort of the space of possible possible embodied minds, right? Any kind of living material, engineered material, software, and patterns from that Platonic space is some kind of embodied agent. So cyborgs and hybrids and chimeras of all kinds. This is, Darwin's endless forms most beautiful, like all the variety of life on Earth, tiny little corner of the space. These things already exist to some extent. We will be living with all of them. And we have to learn to understand beings that are not like us so that we can enter an ethical symbiosis with these beings. And this is why developmental biology and bioengineering is so critical. It's not just the useful living machines. It's not just evolution that we learn to understand, but we actually can use that as a model system to start to communicate with minds that are not like ours. So I'll end here by just saying that improved communication, not micromanagement, but communication and collaboration with living matter is going to lead to definitive regenerative medicine and bioengineering. So everything that you're working on is not just genetic cell biology or biophysics problem. It's part of cognitive science and computer science and vice versa. Bioelectricity is a really convenient interface to that collective intelligence, but only because evolution loves it. It's not magic by itself, and there are other kinds of cognitive glue as well. And bioengineering is an amazing model system for really big questions. How do embodied minds ingress into the physical world? How do they scale and transform? And how can we recognize unconventional minds that are all around us? And I tend to think that that basic diagram of the different sciences is wrong, and that it's really behavioral science all the way down and all the way up. And we can think about different ways to think about it. Matter to life, yes, but also matter to mind first and then life. And we can talk about that if anybody has questions. I actually think cognition is it doesn't come late in life. I think it's a much earlier and larger scale phenomenon, and only a subset of that is life. And then we can even go further. So that's it. I'm going to stop here. Thank the postdocs and the grad students who did all the work, and Josh's team that did all the coding for the MOMBot, and the team in Boston Engineering who built the thing for us. And disclosures, there are 3 companies that have licensed some of the work that I've shown you today, and that provide sponsored research agreements. So yeah, that's it. I'll stop here, and thank you so much for listening.
Thank you so much for your talk. So now we would take questions from the community. I think you can raise your hand, and I can allow you to talk, or you could also write your questions in the chat.
There is already a question in the chat. Do you see this, man?
I do not actually.
Q&A. Otherwise, I read it. Thank you for the
Okay.
inspiring talk. Where is the information on how electric gradients should be stored?
How they should be stored? Well,
Where? Mhm.
yeah, I mean, okay, so they're stored just like basically there are a lot of similarities between how stable electric patterns are stored in brain tissue. They're stored by virtue of stable electrical states maintained by ion channels and ion pumps and gap junctions. It's also similar to how volatile patterns are stored in computer memory. There are lots of models for this. So then we do all kinds of simulations. You could ask a slightly different. So the mechanisms of all this are fairly clear now. You might ask a different question, which is, where did the patterns originally come from? And that answer has 2 pieces to this. The conventional piece is, well, you can think of it like Turing patterns. If you see Turing patterns in chemical media, you say, where did they come from? People say, well, they're emergent from the rules that govern the interaction between your morphogens. That's one way of looking at it. I actually think that's quite limiting. It's okay for some things, but I think it's quite limiting, because in an important sense, you can go beyond this and simply not simply treat emergence as surprise, as something that, well, we specified some rules, we didn't actually know what was going to happen, we simulated them, and now we understand that this is what's happening. I think that there's actually a latent space of possible shapes. And this is not a random grab bag of, as people say, regularities that hold in our world. There's actually a space. This is what I was alluding to in this business of the Platonic space. There are actually specific contents of that space that are that project through physical interfaces that we make. So if you make certain electrical circuits, if you make certain biomechanical objects, if you make or mechanical objects in general, if you make certain chemical kinds of systems, what you're accessing is a space of patterns that can be characterized and exploited, and to make new things. And I think it's really important to have a research program to explore that space.
Okay, thank you. There's a further question. Are ethics programmable, desirable?
Sorry, say that again. Is what programmable?
Are ethics programmable or desirable?
Are ethics programmable or desirable? Wow. Okay. So I mean, I'm not a philosopher of ethics, and so to me, they seem very desirable. It doesn't look that things go very well when there aren't any. That's kind of an amateur opinion. So I think ethics are essential. I think that broadly speaking, what I mean by that is the way that we're going to interact with each other and with other beings that are capable of suffering and joy and everything else. I do think we owe it to ourselves and to everybody and to all sentient beings to develop a system of ethics that allows us to have compassion and sort of positive interaction with each other. Are they programmable? I don't know. I mean, people have over thousands of years have suggested that certain patterns of ethics are contents of that Platonic space, just like properties of mathematical objects. That's a little beyond my pay grade. I'm not sure about that, but I think they're absolutely desirable, and I think we have not just desirable, but we have an obligation to develop systems of ethics that are updated with the new science and not sticking to ancient categories that are basically not going to survive the next decade.
Okay, thank you. There's a further question. Thank you for your insights regarding the anthrobots. Why do these aggregates of cells begin the repair of neutral wounds? Is this programmed into the cells?
Yeah, programmed is an interesting word. I mean, for sure, we didn't program them. We did almost nothing. We provided them a new environment within which they can reboot their multicellularity, and then we discovered that this is what they do. And actually, we're working to discover all kinds of other things that they do. So we did not program them. Now, is it programmed? Programming has when you say program, what you're invoking is a particular kind of formalism from computer science, and that has certain benefits and certain limitations. The benefit that it has is that it reminds you of the existence of software. That is, you can have a certain system that can have multiple different behavioral modes. It's not just doing one thing all the time. That's very powerful. So from that perspective, yes, I think from that perspective, software is a useful way to think about its capabilities. However, it's highly limiting, and I don't think the standard computational paradigm is sufficient for biology, because the notion of algorithms suggests that when you program things, you give it the steps to do the thing it's going to do, and you know what it's going to do, and what it does is exactly what you programmed it to do. I think none of those things are true, I think, even in extremely simple scenarios. So I didn't have the time to talk about it today, but we have simple computational work looking at things like sorting algorithms, which are deterministic, very short, simple kinds of things that, I mean, if anything is a programmable algorithm, that is. And already we are finding that if you know how to look, you discover novel competencies, not just complexity, but competencies recognizable to any behavioral scientist in these very simple systems that we did not program in there, meaning it's not in the algorithm. And we know because the algorithm is only 6 lines of code, you can see it, that there's nowhere to hide, unlike in a biological system where there's always new mechanisms. So I think we have to be very careful with this programmed idea. It brings in a lot of baggage from formal systems in computer science, some of which is very helpful and some of which is quite limiting.
Another question is also, again, thank you for this fascinating lecture. You mentioned injecting the bioelectric signal of making an eye into the gut and its results. How do you identify these signals in a way enough to replicate and reintroduce into other groups of cells?
Yeah, so there's a couple of ways to do it, and we can make certain organs. We can't make all of them, so we have not fully cracked the morphogenetic code or the bioelectric code by any means. But the way we do it is two ways. The easiest way, and the way we started, is to look at natural embryos look at the patterns that kickstart the various organs and try to reproduce those patterns somewhere else. So when you see why are there two eyes and why are they in a certain location? Oh, it's because there's this pattern memory that shows you how to do it. Can we take that same electrical precursor and introduce that somewhere else? So basically take the same prompt that you see playing out in native embryos and put it somewhere else. So that's one way to do it. We now have slightly more sophisticated versions, which are computational platforms that try to predict. That is, if you want a certain thing to happen, here's the bioelectric code you would need to do. You would need to implement, and that is how we do the brain repair. So the brain repair, in fact, it was interesting. We were able to simulate, and the computational model basically simulates ion channels and pumps and gap junctions and downstream transduction events and so on. What we were able to do is after taking that pattern and repairing the brain state, which we had pretty good voltage imaging of what the native brain pattern is like, we were able to then use it on heart, gut, and face. And we didn't have a good voltage imaging for the gut or the heart because those are deep tissues that we can't access optically yet. And so, and yet it worked. So that makes me happy that we can discover some transferable underlying principles that when we learn from one system, it's not just that we can then take it into other systems. But that's what it is. It's a combination of observation using voltage dyes and then computational modeling to try to understand what is it that is really the signaling specificity here.
Okay, another, thank you for the talk. So everybody loved your talk. And here's another question. On a question of language, is it possible to replace the notion of agency with a more deterministic phrasing of moving along different paths in the same dynamic landscape to certain attractors?
Yeah, that works sometimes. In other words, I think I don't think any of these. So you laid out different two alternatives. You said, well, there's this like agentic talk, and then there's this dynamical systems deterministic talk. I don't think that these things are completely mutually incompatible. I think what we're looking at is a continuum. First of all, some of these things are in fact well described by systems by formal models of attractor landscapes. So sometimes that's true, but some of these other systems, for example, they bend their own landscape. They switch between different signals to pay attention to. If you change what you're paying attention to, suddenly you're moving on a different attractor landscape. And so if you bend that stuff enough, then you really no longer are talking about like the tools you're using are no longer really dynamical system tools. They're really behavior science tools, and you can resist it and you can say, no, it's still attractors. I'm just going to lay a bunch of extra stuff on top, and you can do that. Eventually, then you have this very unwieldy set of epicycles that is basically going to try to cash out human behavior as a bunch of attractors. And to some extent, that might work. But I just don't think there's any reason to avoid using the tools we have. We have specific tools developed by computational neuroscientists, in computational psychiatry. We have these tools that are specifically designed to help with systems that are extremely plastic, that can change which landscape they're going to traverse, that can bend the landscape, that are multiscale, where they actually change the landscape for their parts, right? It's just, if you really want to go very reductionist, you can say the whole thing is just Schrodinger's equation, and why don't we just do that? Like maybe, but we have these much more effective tools. I don't see why we should skew low at all times. So I think we have the tools. Let's use them and see where they pay off.
Interesting. Okay, we have one more question, and I think we make it our last, and then we
Great.
can finish the session. A question to wrap my head around this. We can somehow communicate with the alternate intelligence of cells to prompt them to make organs, fix injuries, or tell cancer to stop. And what the Mombids aim to do is be a translator between our intelligence and the cell's intelligence that communicates in their own space.
Yeah, I mean, I'm not sure what the question is. Like all of those things are, I think that's correct. I think what you just said is right. That's exactly right. We are using the bioelectric and other interfaces to communicate with the with a collective intelligence of cells and tissues and even molecular networks. And our goal is to communicate with them and to navigate their space with them, so we have some idea of what it's like to be them. So it's a smooth transition between these kinds of projects and really the big questions of mind and embodied minds and consciousness and so on.
Oh, okay, I think the last one was really only a clarification question and want to hear rephrasing.
Great.
So, Mahin, do you have something to say at the
Uh,
end?
no, I think actually there's a lot to think about. Thanks. So yeah, thank you for your talk. I think the classmates also said they were very happy with it. And if nothing else, then maybe we wish everyone a happy end of the year and a good start to the next one.
Thank you
There's,
very much. Thank you, everyone. Bye-bye.
Thank you.
thank
Bye-bye.
you. Bye.